All right, so we're going to go ahead and get started. You might be a few minutes early, but that's quite all right. And welcome to this, your finest and final hour of the day of programming. And what lay ahead is the Network Bash, and I know you're all looking forward to that. And I just want to mention to the sponsors of the Network Bash, it's Elevate Bio and something I'd never heard of, which is the Money Channel. NYC. And I'm thinking the Money Channel, I think, oh, we all want that on our cable system is the Money Channel right now, so good for them. What we're going to do today is in this last session, it's going to be a collaborative conversational piece. I'm not going to stand here, I'm going to sit here, and I'm going to let them do most of the work for me. And we've had prep ahead of time. And as you know, the topic of this session, and every time I read this title, I come up with some different nuance as to what it actually means. But if I'm looking here, it says comparability to continuity, aligning late-stage manufacturing changes with global post-approval control in cell and gene therapy. So we're going to leave the cell and gene therapy piece in there, and we're going to back up and we're going to go to the late-stage manufacturing changes, because I think that that's where we'd like to spend some time, and comparability. Has anyone heard of comparability here in the room? It's okay, raise your hand, that's good. You know, it's kind of a pretty common theme and a common thread. We talk about it a lot. And so what I'd like to do is introduce myself first. So I'm Don Fink, and I'm Master Practice Expert Regulatory at Dark Horse Consulting. And as you may know, if you've been following the group, we've expanded, and we've acquired two groups, Biotech Logic in October of 2024, and most recently and excitedly, we have Converge Consulting in 2025. So we are, in fact, a group. And today, this topic of this session really represents a microcosm of what we mean when we talk about comparability. Because comparability is a very extensive and broad and consuming topic, we're going to focus in some areas specifically, and we've arranged to do that. But it's inescapable. It's an element of all of our cell and gene product development. And just as a companion to this session, if you are looking in your program ahead tomorrow, there's also something called the Science Slam, Comparability Studies Walking the Tightrope. So this is a plug for them. So whatever you don't get from here, you can go and follow up and have from them. All right, so what I'm going to do here is real quick. What I want to do now, I'm going to switch to my type notes. I'm going to allow myself to introduce this really fine panel that sits before me. I'll get through right now their names, and then we'll come back to them in just a second. So immediately to my left is Ignacio Núñez, and he's Chief Operations Officer at CellReady. And then I have Sousa, who is also Chief Regulatory Officer. She's at Aspen Neuroscience. And then Philip Willits is Chief Commercial Officer, Cell and Gene Therapy at Catalent Pharma Solutions. And finally, I don't want to say finally, at the end, because she's alphabetical, is Annie Marie Woodland, and she's Senior Vice President, Regulatory Affairs and Quality with Beam Therapeutics. And the thing I wanted to point out that you may not know, unless you haven't looked, we have two innovators and two contract manufacturers. So we have a little bit of a balance here, and we'll see how that plays out later. All right, so the palette of manufacturing changes that are often made during product development, I think we're pretty familiar with. It's not included, but not limited to. We have raw materials quality changes. We have sourcing that we need to do, rejuvenation of capacity-limited starting materials, such as master cell banks, working cell banks, operations modifications to accommodate, scaling, we'll have to think about that. And then there's formulation changes, adoption, implementation of new analytical methods, changing of manufacturing facilities, and all of these things, to some degree or another, come into play, and comparability is an element of some of them, not all of them, depending on the risk assessment. Late stage, and here's where we're going to define that. So what we're talking about, late stage manufacturing changes, these are things that are made immediately prior to conducting a pivotal study anywhere. So you know we have compressed timelines now. Anywhere you'd want to start collecting clinical data, to me that's pivotal, because you're going to use that data to support your license. So a late-stage manufacturing change, anything before a pivotal study is going to be done. Then sometimes when we see it more often than I'd like, we see manufacturing change actually implemented during a pivotal study. There may be a reason for that. Something couldn't get around to it. They wanted to get started, but need to bring the new facility on. That happens. And I think the one that is the most concerning, is the most difficult to deal with, and this is all prior to the BLA itself, is when all of that data, all that efficacy information has been gathered, and then they come in and they say, okay, here's our commercial manufacturing process, and this is what we're going to include in the BLA. How is that going to work? So those are the three where I'm thinking about late stage changes, and that's what we'll do with that as we go through this conversation. All right, so I'm going to stop now. You've had enough of me. What I am going to do is I'm pleased to be able to introduce to you my panelists on the floor, exceptionalist folks. We've gotten together and we've spoken and we've had reasonable conversations for the most part, and we're going to leverage that today. And what I'd like to do now is I'm going to give each of them an opportunity to introduce themselves and make a statement or two about their, what I call their close encounter of the third kind, first kind, or any kind related to comparability, whether it's been in doing a protocol, whether it's been contemplating the need to do this, or whether it's been executing within their manufacturing facility to do that. And while they are doing that, I'm going to try something a little trickier. I'm going to have our two folks from ARM who are part of here. They're going to be up front at the ready, and we're going to have early bird questions. Okay, so by early bird questions, what I mean by that is if you wait to the end, you may not get a chance. And you may have something you really want us to comment on or ask us. And so once we conclude with our wrap up with these introductions, you have first two to the mics. That's all we're going to have. But we'll have two early bird opportunities for folks to ask questions. That'll help to also get our going. All right, so I'm going to go ahead and start with Ignacio, and then I'm out, and go ahead and introduce yourselves.
Okay, thank you. So my name is Ignacio Núñez. I'm the Chief Operations Officer at CellReady. CellReady is the first G-Rex-centric CDMO in the world, and our vision is to structurally reduce the cost of cell therapies by identifying and applying standards and unlocking the full potential of G-Rex. Full disclosure, I'm not a regulatory expert. I mean, I'm an operational user. So what I can provide is, hands-on experience by dealing with developers in multiple stages in the road towards commercial manufacturing. Now, to me, comparability is not a regulatory issue. It's more of a operational discipline. So what is the meaning of that? Well, so if you don't deploy good standards, if you don't have predictability in your processes, if you don't have good control in your value chain, eventually your comparability study becomes a reactive fire drill versus a proactive risk management. Look, how many times have you seen that you are in the lab and you said, Jack is our best operator. He's got the touch. I don't know how he does it, but he goes there, he's got the best performance, right? It's like, oh, that's great. Kudos to Jack. Well, that is the definition of tribal knowledge. The tribal knowledge is really, really dangerous because that means that somebody very likely is not following the batch record, which, by the way, when I come to these events, very rarely I see anybody talking about batch records and master batch record, which is really the base of how we do science in a controlled environment. And then you have all that variation simply because you don't have a good standard or the standard is not followed. That means that you have a discipline issue in your operations. Look, when you look into the best supply chain in the world, you go to big pharma, you go, for example, to Johnson & Johnson, they deployed what is called the Johnson & Johnson operating system 10 years ago, which was a major, massive exercise to deploy standards across the more than 50 manufacturing facilities all around the world. And what they will do is they will enable that when they want to transfer one product from one manufacturing facility to the other, will be much easier simply because the standards are somewhat similar across the board. I really miss a good standard in the cell and gene therapy field. When you really look under the hood of most of the processes, you realize, especially in cell therapy, that once that you choose the platform, your manufacturing process is 80% exactly the same. You have maybe 20%, which is your secret sauce, but for the most part, everything is exactly the same. Yet people really keep the process like it's mine, but if you really share it, you will see that actually you have a standard that we could, as an industry, continue refining and improving and enhancing. And eventually, to compare will be really, really simple because you will be talking about just comparing that 20% variation versus the 80% standard. And then the last part, and I just close the opening remark, is just, look, this industry is so new that you have continuous improvement methodologies that help you to continuously improve, such as Lean and Six Sigma, that are all about the standards. And by the way, the agency absolutely loves companies that are using Lean and Six Sigma because that is a guarantee of predictability, standardization, structured problem solving, having things under control. And there is almost no penetration in the field. And I would like to do a test, if you allow me.
Absolutely, go right ahead.
I would like to ask the audience, how many of you are familiar with Lean and Six Sigma? Could you raise your hand? That is impressive. That is almost 60%. It's blowing my mind. Okay, how many of those are actively applying Lean and Six Sigma in your organizations? That's a different story. That's less than 10%. There you have it, and it's for free, right? And what those methodologies are gonna tell you is deploy standards in your value chain, end to end, become predictable. And when you become predictable, to compare is really easy. You have to understand that when you have a value change, is task after task after task. The previous task carry a value to the next task. So if you are not predictable, by definition, you're gonna catch somebody along your value chain off guard. And then you have variation. So when you want to do comparability, then you get yourself into trouble.
Thank you, Ignacio. All right, Anna, to you. Close encounter of any kind.
So Anna Sousa, I'm Aspen Neuroscience, and I am Chief Operations Officer in Quality, and I agree with everything that Ignacio has said, but I also think it is a regulatory challenge because I think a lot of times we don't bring the FDA along, and then we show up with a surprising comparability plan that is not agreeable. I think one of the things that I struggle sometimes, and I make sure that we think about a lot in the organization, is when along the life cycle to make the changes and what changes. And I worry about, compounding changes and what that's going to do to your product. So we work on iPSC cell-derived autologous cells, and I agree, a lot of the process has been established. But we also have a lot that we have introduced, and we're doing a lot of strides to actually automate a lot of our processes and take that tribal knowledge out of the day-to-day. So I think it is a regulatory. I think it involves a lot of quality. You can't just do comparability and automate in manufacturing. You have to think that comparability needs to include the quality automation as well, because otherwise then quality becomes your rate limiter. And I think you need to make sure that you bring the FDA along and that you establish a program that's going to be able to meet the needs. I think someone was saying that at a different venue today, start with the end in mind and consider the impact of your comparability on your life cycle, because it's not only about CMC, it goes way beyond. Whatever decisions you make, including pre and post change, is gonna have impact on commercial, it has impact on payers, it has impact on clinical. So it's way beyond CMC, it's way beyond manufacturing. So those were, I think, my comments.
Phil.
So yes, hi, Philip Wills, Chief Commercial Officer at Pharma Solutions. For those that don't know, Catalent, we're a global CDMO that does a broad array of different type of product types. One of our specialties is in the cell and gene therapy space. We have manufacturing facilities in Europe and in the US for cell therapy. We have a gene therapy manufacturing facility on the East Coast of Maryland in the US. We also do plasmid and iPSCs as capabilities. In terms of my close encounter, they said it's, hearkening the discussion that you're mentioning for tomorrow, the walking the tightrope, right? So it's, very much as a manufacturer, you're trying to improve the process. I think also as a sponsor company, you're also trying to improve your product. But then you have that back of your mind, that FDA edict that if you change too much, it becomes then a different product. And what does that mean in terms of having to do more trials and things like that? And so we had an encounter where we had an asset that we had manufactured phase 1, done some process intensification that actually worked really well to the point where it was almost like, well, maybe we need to step this back a little bit because the product is too much better than it was when it went through the phase 1. So I think that really is a tightrope of trying to improve your process, trying to improve your product, all the while making sure that you do stay comparable to the original IND filing.
All right, thank you. And as we go to Ann, I give 2 folks, early birds, get your questions. If not, we'll just jump on. Go ahead, Ann.
Anne-Marie Woodland from Beam Therapeutics. I'm the Senior Vice President of Regulatory Affairs and Quality. I also agree with Anna that it's not only a CMC issue, but it's also a regulatory and quality issue. I think it's very important to have that continuous planning as you're going through your product life cycle, and there's always going to be optimizations. I think comparability can be very unexpected and very expected at the same time. And I think it's very important in order to, as you plan your comparability protocols, and it really to know your end, but where you, as you're going through, what do you want to, how are you going to sort of change that narrative, and working very closely with the FDA, I think has been game changing, and really bring them along to what you're really trying to do.
That's great. Do we have any early bird volunteers? Yes, Sarah, please.
Hi, Sarah Mills, Artiva Biotherapeutics. A question when on the therapeutic developer side, I think and Don, we're starting to weave a very fine needle. With the most recent draft guidance that came out regarding RMAT, FDA is starting to codify that they don't really want us changing the process after we get an RMAT designation. And thinking about how early on the developer side we're looking to get an RMAT designation, and then obviously, as everyone on the panel has really spoken to, the continuous improvement that all developers are looking to do throughout development of a therapeutic candidate. I'm wondering how everyone's thinking about that and life cycle management of an asset as you move into later stage development. Thank you.
That's a really, really good question, and I hadn't thought of that until you mentioned it. So innovators first, because they're she's in your space. The question of, so I don't want to speak too much because I'm not on the panel, but with the RMAT, as you know, it's based on clinical evidence it's enough supportive to say this is likely to have probable benefit. And having been on the FDA review team as a CMC reviewer, the only question I'm really responsible for is, does it meet the definition of an RMAT product? But the clinical reviewer wants to know what version or versions of the product have been tested or will be tested, and if this data is to be able to give that RMAT designation, and you go ahead and manufacturing changes, can we still rely upon it? So how do you have you thought about that, and how do you both Anna and Anna, how do you think about dealing with that?
I think that's where you have the end in sight, where you have, it's really optimizations that you focus on and not completely changing your product or your process. And it's really, as you're starting from that very early phase 1 trial, your manufacturing is just not going to be as sophisticated as it would be in even phase 2 if you're in a rare disease or you're on an accelerated pathway. So it's really being able to refine and optimize so that product is not different.
Yeah, and I think also is it is a challenge, right? Because you do want to get RMAT as soon as you have preliminary evidence of clinical benefit, and you can achieve that very early, and your product is not yet optimized. I think for us in cell and gene therapy, is we have to focus very early on your CQAs and your CPPs and really understand your process, and then have robust systems that could actually be able to demonstrate to FDA that, yeah, I made X process, but I could still maintain that this is the same product and that it actually has no adverse impact or effect. And if you could demonstrate that, I think you could make a good argument to the agency that it is still the same product. I think the FDA in the past has, although they wrote that in the guidance, the same like in the comparability comparability for cell and gene therapies, I think line item 46, they recommend that you improve your product, but then on the next sentence they say, but be careful, don't improve it too much, because then it becomes a new product. So I think we are walking a very tight rope, and from my perspective, it's all about really understanding your product and really having strong CPPs and CQAs early on in order to be able to establish comparability and have a strong conversation with FDA that whatever improvements you are doing, as long as you show that you have not adversely impacted the product, that you can stay and keep your arm at, but it is a tight rope.
So contract manufacturers, do you want to get involved in this at all?
I mean, we definitely believe in the concept of trying to have the end in sight at the beginning, especially if you're going for, some accelerated approval pathway. And I think that's where you can leverage a manufacturer, certainly one that has had experience with previous late-stage products, to being able to give you guidance on how to start that process at the very beginning so that you don't have to make a lot of unnecessary changes, right? So the idea is to set yourself up where what changes you do make are purely based on your product, based on the unique circumstances as you develop it, rather than picking a suboptimal process to begin with that's going to need to be improved before you get into manufacturing for commercial.
Thoughts? Are you good?
Well, I wonder how much the feedback from the agency is really related to your product and how much is related to the platform to manufacturing your product. Because there are platforms that are extremely complicated, right? And the agency is going to tell you, look, you got it, don't touch it, right? Because it's really complicated. You have so many parameters that now that you got to fine tuning, just you better not touch it. But you have some other platforms that are more nimble, flexible, faster. You can test multiple conditions, and you can show comparability and easy changes to the agency very, very fast. So I wonder how much is really product related versus really the wrong platform or platform related, if that was the one that you chose at the beginning. And to your point at the beginning, right? So think with the end in mind, but the decisions that you are taking early on, the platform may affect your path towards your commercialization.
Sure.
And when you say that, may affect your path, this becomes the continuity piece. All right, so continuity here, what I'm thinking continuity means is if you can't do comparability well, you're not going to have a very continuous or good continuity in your development process, because you're always going to have to be stepping back and reworking and jumping out. And so I think that that's important. Thank you for bringing that about how that's important. I want to and I'll just reflect back to the original question and a little bit about what it might mean. I think if you get the RMAT and you have the process, and this is the product you've done, then you start iterating changes in the manufacturing process, and now they have questions about, is it comparable or not comparable? Does that jeopardize the RMAT? That's what I'm wondering. And I don't know the answer to that, but I'm just wondering that. Do we have one other question before? All right, good. We have a second early bird. Thank you.
I wanted to go back to the tribal knowledge and trying to help establish what controls you want at the appropriate time as a team. And that includes the person from the FDA who's the reviewer. Because isn't that the time when you find out where the most difficult teaching has to be done? Whether or not it's the person who worries about the clinical trial, if it's the FDA reviewer who's not had a lot of experience in a certain area, somebody has to take the lead on being the person to determine what level of controls you're going to have and be able to communicate that. Otherwise, I think you get yourself into this losing sleep at night because I think I'm okay, but what happens and so on and so forth. So I get this pushback from people I talk to where they're always afraid, they think they can't talk to the FDA because they're going to always go back. So I come back with a simple thing, and I said, why don't you come up with a dashboard? and say what's going to be on the dashboard early on and try to get some agreement on at least the dashboard as a way as you start looking at a strategy for CQAs and CVPs. That's the only thing I wanted to bring up now, because I think there's so many people involved that don't want to do the wrong thing, but nobody wants to be the one that challenges what level of controls do you need. Yeah, for the whole team, for the whole team.
So for my two contract manufacturers, I'll start with you guys, and actually you did raise this element of tribal knowledge and what that implication is. So, and you talked about standards, so just maybe, again, kind of revisit that in a little bit about the context of the question as you heard. And then, Philip, if you have anything to add, you're welcome to.
Sure. Gonna go first?
To me, the standards in a manufacturing facility are the law. You better not break them, because that will have consequences, right? And you can do it through dashboards. You can do it through reducing the degrees of freedom, right? You have to definitely provide visibility to your standards, but so what do I mean with the standards? So you have obviously the master batch record, right? You have your pick list, you have your assays, right? Your hub, the way that you gowning and degowning, you have your scheduling, right? Meeting your scheduling, because you have the interdependencies. So you have a specific schedule, you're gonna have QCM, you're gonna have QCA, you're gonna have operators in and out, right? So your scheduling and your standard becomes a heartbeat of the manufacturing facility, right? And when your heartbeat is predictable, your body works perfectly fine. When your heart is not predictable, then you have an arrhythmia, right? And then you are in trouble. So to me, the way to have visibility is if you start deploying standards, stabilizing every part of the organization, every time that you break the standard, you see it right away, and it's really easy to correct. But if you don't have a standard, you live in continuous chaos, right? And then you live in a place in which low, medium, high priority does not count. You are living continuous firefighting, right? And that is really, really hard to come around, right? So yeah, so to me, it's deploying standards and making the standards in your operations the law.
I agree 100% with that, but I mean, what I would also point out is that there is an advantage to working with a manufacturer who's done previous products before, because I mean, some things you learn just through going through the process, and you realize that initially when you write up a protocol or write up your batch record, you think this is clear. It's only then as you start to see those people implement it and execute it that they say that maybe there's deviations and things like that occur because it's not clear enough. And so those are lessons learned that can help the sponsor companies in terms of how to really standardize the language in the batch record so that it really is not tribal knowledge and really is a proper recipe on how to execute.
Yeah. And so now, innovators, do you have any play in this space? So they're really talking about, because often these start with you, these manufacturing processes, and they start with whatever approaches you're using to try to keep them within control and rigorous and that kind of thing. So how does this play out in your context? And I'll go ahead and start with Anna, and I'll come, because I know, Anna, you got something to say, so I'll give Anna the first shot at it.
I think reproducibility and being able to demonstrate reproducibility, so having your standards be the law, I think, is really how you get to that point. I think your assays also factor in quite a bit, how robust are your assays? Are they reproducible? There's a huge focus on validation of those assays. And that's the only way you can really judge if your manufacturing process is also reproducible.
You talk a little bit about your manufacturing processes. I know it's a unique kind of endeavor. So.
Yeah. I agree with Annie. I think for us at Aspen Neuroscience, aside from us automating what has been in the past, a challenging manual process, but we also trying, and we have actually accomplished using machine learning algorithms to take all the tribal knowledge out and have machines really replicate what in the past was the decision-making of an operator. The machine will do that. When to add reagents, the machine will do that. So the machine is actually going to be just executing what an operator in the past will be doing it, but it is doing in a automated fashion decisions on what cells to pick. So we take all the chaos and that decision making. We are humans, and sometimes you're not sure, right? And you're just like, oh God, I'm tired, or is this the same cell behavior I saw before? And the machine will have that data to be able to make that decision. And to the point that if there is any challenges that, we'll have alarms that then we'll bring an operator to confirm or deny the decision that was made. So I think also having artificial intelligence included and training, I think would be really helpful and take all that chaos. So it's automating some of the things that have been done in the past by our operators and automating. We are just doing a lot of strides on that. We're operating 90% of our process, of course, with our collaborators, and we're really excited of what we're doing and where we're going for our pivotal study. So exciting stuff. So that's kind of how Aspen, that's our take, is to actually use machine learning. And this was something that started, speaking about starting with the end in mind. When Aspen was founded, this came from, this machine learning had already been the concept that was transferred from Scripps Research Institute that came with the scientists to Aspen, and we've just been continuously improving in that. And what we're doing in that space is amazing. So I'm really excited for patients and for the consistency and the quality that we're bringing to our product by automating our processes.
Now I'm gonna give you another chance. I want you to hold that question. I'm sorry, I only want 2 early birds. Okay, I'm gonna hold that question, but when we get to a couple minutes, you come right up, all right? Don't forget that question. All right, so we've been working at Dark Horse. I don't want to beat a dead horse, but I'm gonna do that. And I had as my opening gambit, and I want to give it to you in quote because it puts it in perspective. And we're all talking about the comparability guidance, which is, if it's not your Bible, it should become pretty close if you're in the comparability space. But what we've been referring to, and then I'm going to go down the line and just let you react to this question. And you have to some extent, but we'll try it one more time just for my sake. Thanks. So in the guidance, it's on page 2 of texts. It's on lines 46 to 49 out of 880 lines in this guidance document. And it says, and I quote, "We note that while improvement in product quality is always desirable and encouraged, if the results of comparability studies indicate an improved product quality suggesting a significant benefit in effectiveness," and I'll give them this, and/or safety, okay, but if it's an improvement in safety, well, then the pre- and post-change products may be different products and therefore not comparable. So my question to the panel with that statement, which I've heard should not have been included, I'm asking you this question, how does this statement, from each of your perspectives, thinking late stage, having the end in mind and already looking ahead, how does that statement affect your enthusiasm for introducing a late-stage manufacturing change during product development prior to submitting a BLA? And then it could even extend to post-approval, but we're taking that off the table today. What would be some practical implications for you in each of, we have contract folks and we have innovator folks, in response to that statement for making a late-stage change? And we'll just go down the line. Your reaction to that statement?
Well, don't we have an ethical dilemma? Because, I mean, obviously, we want to give the patients the best shot, right? So if we have system that is stopping us from delivering the best therapy to give them the best chance and hope, I think we are really wrong. They're really wrong, right? And then what I will say is like, well, if still we want to walk the process, I mean, we should pick a platform that enables that, if that is the case, we could do comparability studies in a very accelerated manner, rather than becoming a huge burden that takes months and millions of dollars, which for the most part, when you choose the raw platform, like full end-to-end automation or something like that, you will have to go through. So I think those are my 2 comments.
So time and money, yep, very important.
I think it makes you think twice, for sure. But I think, and I agree with Ignacio, that should not deter you for doing the right thing. And I think you need to just bring the FDA along. I also think that data is king, and I support that you need to make a lot of process development before you engage in actually establishing a comparability protocol, to really ensure that you're not adversely changing your product, and then let the data speak for itself, right? And then you could come to FDA and have a conversation. In general, I think that the FDA, if you show them that your data and basically your product is comparable, and that you understand the challenge sometimes with cell and gene therapy is that the depth of the knowledge of the product is not tremendously deep, and it is double-edged sword, because you could really understand your product to the single cell level, and that could be also a double-edged sword, right? But I think you just do the best, and again, with the end in mind, thinking of patient, thinking of quality, and really understanding your process, having strong and robust assays and constant communication with the FDA so they come along with you and understand your journey.
Yeah. And Philip, I'll just kind of help you too a little bit with this as well. So let's say Anna has this manufacturing process and wants to make a late-stage change. You know what this means, what that statement is, and she comes to you and says, what can you do for me? How is that going to shape your approach to helping them set up that process in your facility?
No pressure.
So you mentioned enthusiasm, and what I will tell you is that statement has definitely suppressed tremendously the amount of enthusiasm that the sponsors, are willing to take in terms of making a change. So, I mean, we often see something that comes in, we see the process, we see it's suboptimal, we say, you should think, so say on a viral vector side, right? So, I mean, there are a couple of things. One is, oftentimes I use centrifugation as a way to kind of purify the empty versus the full. In terms of manufacturability commercial-wise, that's gonna be, you talk about tribal knowledge, you talk about reproducibility, pulling bands from cesium chloride gradients is not that, right? So, you talk to them and you say, well, we can with chromatography get the exact same critical quality attributes that you were seeing in your phase 1 material. You know how many times they say, no, we're gonna just stick with what we got and this is what we're gonna go with. So the enthusiasm is 100% suppressed. Yeah. So also the other one was going from adherent HEK293 to suspension. So even if you take the same exact adherent cell line and make it suspension adapted and use that, again, no enthusiasm. They say this was our process, this is what we're staying with. We'll tackle that post-approval is oftentimes the response.
And the only advantage I can see to that is at least they have an approved product to give from which they can compare their comparability study, but that, it's going to be a big lift, isn't it?
Yeah, again, I think end in mind at the beginning and as early as you can, try to implement improvements to your process.
Yeah. And just with you, Anne, because I know that you're dealing with a rare disease population, you're dealing with limited sets of patients and manufacturing experiences, so there's probably some additional challenges there for you as well.
It's definitely a fine line of over-improving the product and not understanding the over-improvements. And really, you have to be incredibly objective about what you're trying to achieve, especially with a rare population. And if you're changing it too much, where your analytical comparability can't really give you the answers that you need, you're now eating away at, to having to almost redo a clinical comparability in a patient population that's rare. So I think you really do have to be incredibly objective and really understand, is this an optimization or is this a next-gen product? And I think that's And if you know, it's really about knowing your product as well.
Yeah, and for my money, I think it would be helpful to just change that sentence to say, if they don't, in fact, are different enough, it's just that they're not comparable. They're not a different product. They're just non-comparable versions of the same product. And then you would have to work within that paraphrase. Yeah, that's okay. So good news for you, we're going to get to jump way ahead just for funsies. And I'm going to let them have a good roll at me, because I'm a simple guy around comparability. Most of all, what I'd say is I don't believe you can do it before you get a licensed product. I think all of this is magic because you don't have a place to start or a place to finish. You're going against unknowns, and you're having to create a universe of data that may or may not be representative. So my view, and I'll read it this way, so I'm going to throw down the gauntlet here, and we're going to go down the list. And irrespective, in my view, irrespective of the hierarchical granularity applicable to establishing suitable Tier 1, Tier 2, Tier 3 attributes for the purpose of demonstrating, and I'm using analytical comparability here because we're trying to avoid having to do additional preclinical or clinical work. I would argue that there's really only, you're going to, all of these attributes, there's only one. There's only one critical quality attribute that you need to protect, defend, and guard against and not have change, and that's potency, or your potency assurance. Because if that doesn't change, I don't care what anything else is done. If you're good with potency. So, and I think if you can defend that rigorously against your equivalence margins, you've reached an acceptable determination for me. So tell me why that's wrong.
I think potency is very important, to know that your product hasn't changed and that you're delivering the same product that you've delivered in your clinical trial. I think there's a constellation of attributes that define your product, and I think those are, may not be equally important, but I do think it really does shape what your product is and how you've characterized it. So I think potency is not the only answer.
I'll also say that potency assays, there's oftentimes one of the things that the sponsor companies maybe don't spend enough time in early enough. It's something that comes in as their mid-clinical development, and then you don't have the history and the understanding of your potency assay to really be able to judge fully whether or not this is comparable or not comparable. And I think just the space itself, the rare disease space, these are complicated mechanisms of action with a lot of extraneous things that could be going on, and having an assay that is truly predictive, I think, has been a challenge in this space.
Yeah, and I certainly agree with the singular nature of an assay, or any one assay is doing that. And I also would encourage, or I think that if we can get agreement with the agency about what our potency assay strategy is, that's more defensible than it is to not have that. But you're really right. Back in the early days, it used to be, when do I need a potency assay? By phase 3 when you start your efficacy studies. And it turns out, no, you need it like from day one, because or at least an activity assay that you can judge your material by to make stability and changes, so on. So, all right, I'm going to start with Anna for her last, because she's really going to scrub me. Go ahead, Ignacio.
No, I agree with Philip. I think we have, clients that come with potency assays that are really, really substandard, and they don't realize that they're going to get in trouble later on. So, I mean, it's good if they will be open to be coached, say, look, it's coming, so you better do it as soon as possible. But yeah, for the most part, I agree with Philip that you see clients not taking the potency assay to a level of standardization that they're going to suffer later on as they progress in the clinical trial.
Yeah, I think potency is very important, especially early on. But I don't think I disagree that it's the only thing that's important. Especially, it is important because that means you understand your mechanism of action. And that is really helpful and important. But I think that there's so many aspects of your product, particularly in cell and gene therapy. And I agree with the constellation, but there's purity, there's viability, there's impurities that it could be stability changing. So there's so many aspects. The constitution of your product, what cells are in there, are they changed? Are they the same cell makeup that has an impact on potentially efficacy or the safety? That, I'm sorry, I hate to disagree with you, that potency is the only thing.
Yeah, that's only if your potency assay is bad, right? All of that can work if you have a good potency assay or potency assurance. But if not, I see what your point.
That's not quite.
All right, so I'm going to lose that one. That's fair. All right. We are actually at a point where now, I'll come back to my questioner, And you are, get to start us off. Come up with that question, and then anybody else, we have about 10 minutes here for some follow-on questions from the audience. Go ahead.
You mentioned earlier that you use machine learning to figure out which operators are doing activities good or bad, and the machine then learns to make which decisions based off of, I presume, someone telling them that this operator made the right choice. Could this same strategy be used to determine your critical process? Which steps are critical, which steps aren't, based off of whether or not it mattered if the operator was good or bad. Basically, can machine learning be used to figure out your CPPs and CQAs?
So I have to clarify, and maybe I wasn't clear, what we're doing is having machine learning make the decisions, not the operator. So we have used we automated what the operator decision would be, and we've trained the machine to make those decisions in those situations. So I think that what we're trying to deliver is to make sure that we take what Ignacio was mentioning, the chaotic decision-making or the tribal knowledge out of it, because we've trained the machine with all the tribal knowledge, and we put it in the machine learning, and the machine will make the decisions. What happens is if there is at any point that the machine can't make the decision for any reason, then the human intervention is brought in. Does that answer your question?
Sounds like that. It means you only allow the machine to feed off or learn off the good decisions.
Correct.
So it's the data you feed in, and then
Of course.
and
It's
what you're
always
training
about
it
the
on.
data. Machine learning is
I
about
see
what
a
you put
question
in.
hand up over here.
May I?
Oh, sure, absolutely.
Just
While
one-
they're waiting, go ahead.
So Anna, one question. So what type of decisions would you ask operators to take in a GMP setting?
You're not supposed to ask me questions.
Well,
Okay, a follow-up question. So, Anna,
so maybe-
what kinds of decisions?
Maybe it's another question. I apologize. Look, I think operators shouldn't take any decision at all, because maybe 1 out of 10 is the wrong decision. So I think your level of standardization, and look, it's possible that you have a process that, it has some complexity, so some decisions are taken, but when you are in a GMP setting, you are an operator, you're being 6 hours working the process or whatever, and then, the batch record asks you questions, and you have to make calculations, and you have to make decisions. That's a recipe for disaster, right? In my mind, operators should not take decisions at all. At all. Everything should be really, really clear. Everything should be like a formula or something like a sort of workflow, but no decision whatsoever. So
I agree.
that's why. Sorry, I want to put you on the
You'll talk
spot.
about that offline over at the networking bash. Okay. We have a question. Yes, please.
Yeah, so one of the concepts in the ICH Q8 is the design space, and that you, the sponsor, propose your design space to the agency, and that operating within your design space is not considered a change and does not require you to, file an amendment. So I'm curious, in early pharmaceutical development, what are some best practices that you would counsel developers when you are defining your design space?
That's a really interesting question. I'm thinking, Philip, you guys have a fair amount of experience in at least trying to create design spaces when you have a number of innovators coming in and asking you to do manufacturing tech transfers with them, and you need to create a place where you can still do some, process optimization, but not trigger some outside consideration. You want to maybe give them an example of that?
I don't know about example, but I mean, certainly, there's, you have the backbone of your process, and in that backbone of that process, there is room for which you to tweak things and to get it into a more reproducible and optimal expression levels. Again, where you start will affect, what that space looks like. And again, I think if you can put your initial process much more within what is your end in mind goal, your then tweakings aren't going to create, a vast difference in terms of your product that you won't have, as big of comparability issues.
And so, Ignacio, I was thinking also some things that we commonly do is, and I think it's in the guidance as well, if you're going to introduce some change in a process, they ask you to do risk assessment. And one person's risk assessment is another person's risk assessment, and which eye of the beholder. But I think that that's a process that you may look at when somebody comes in and asks, we'd like you to take our tech transfer and help us with this manufacturing, and you want to introduce some things. What kinds of risk assessment pieces do you look at in that way? And anyone else after this can jump on that.
Well, I think it's related to the way that you, for example, filed your R&D and how specific you want to be and how much you limit continuous improvement. Because, look, I read R&D in which, for example, you have, let's say, a Mars bars or a Lobo machine on one side of the room, and then you wanted to move it to another side of the room, you may have to file an amendment, right? At that level of ridiculousness, right?
I hope not. Has it happened to you? Okay.
Yeah. So I think it all depends how the levels of flexibility that the client brings. But at CellReady, we are aiming to be a center of excellence. So we are going to always apply continuous improvement, because without really having that license to apply continuous improvement, improve the workflow, improving the master batch record, remove as much as possible the decision making in operations, then you're going to live in continuous firefighting. It's just really a fact, right? So we try to do our best, and we are really stubborn. That's why we really filter the clients that we take to CellReady, because they have to be open to be coached, and they have to be open to apply continuous improvement. Otherwise, what is going to happen is we're going to be the ones suffering through, and the capacity is not going to be there, the variation is not going to be there, the costs are going to go to the roof, you're going to have all kind of quality issues, you're going to have all kind of non-conformances, and eventually really is a recipe for disaster. So you have to reduce the degrees of freedom as much as possible, and everything is started by having the freedom to apply continuous improvement to substandard processes.
And just to thank you, I think that's great. I think and if we can try and help one little bit more, I think what he's trying to do is can we create an actual manufacturing space, design space, where we can iterate these changes and not have those changes fall out such that we have to make now a new submission or a comparability exercise. We can simply say we are within a space and these results are okay, and we don't actually need to do comparability. Is that something that you've tried to approach or do, or how would you even try that?
I think that there is a space for that. I don't think it's in every area. I think once you talk about specifications, I think early on there may be a space, but I do think if you think you start with the end in mind, you will start thinking about those things, that if I move the machine from A to B, that's not an amendment. If I am going to change my operator from C to D, that is not an amendment, right? So you have to write your documents that allows that there is some flexibility and that there's a range of things that you can demonstrate that there's not impactful, and therefore you're not going to have to do a protocol or a IND amendment all the time. But I think it takes a lot of forethinking and experience, and I think perhaps this is where the experience that these folks could bring, right? Because they've seen it all and had a lot of experience. But it's about, for example, at Aspen, we're a small organization. We have end-to-end. We have a very complicated process, a lengthy process. So we do think about those things all the time, and we do surround ourselves with consultants that help us think these things through, and have risk assessments that are very well thought out, that we turn all the stones to try to be able to say that it's flexible enough but robust enough to be able to stand the pressures of development, the life cycle, and comparability. Also, one of the things that we don't want to do is to have a comparability process or a protocol that every time you need to do an improvement, you need to put a new protocol in place that's completely different. Or you need to do a new set of studies that you've never planned for. And again, thinking about the impact, right? You don't want to have to do a clinical bridge, right? You don't want to have to rely on animal studies all the time. So I think the thinking of and having help of consultants that've been there, done that could help you, think it through what the risks are and what the range is and the flexibility, and not think that you have all the knowledge in your company, because we all have different experiences, and we've seen some of our unfortunate colleagues or companies that have challenges, right, that showed up with either a new process or were on hold because they didn't report, right? And we should learn from each other.
Yep. Anything you want to add to that, Anne?
I think the design space is good early on. I think as you get later and later in your clinical trials, it's probably more difficult to have that flexibility. But I agree. I think, having an IND that's very flexible early on, and then basically start, locking things down as you have that product understanding is much easier. And then your change management procedures will handle a lot of anything that's not covered in the IND.
Great.
I will add one other thing,
Please.
and that is, oftentimes that manufacturing design space of what you can and can't do, traditionally, historically speaking, gets really fleshed out and solidified as part of process characterization work, right? Sure. But one of the things that I'm seeing with these, rare ultra diseases where they get the fast track designations and they're moving fast, is that they don't want to spend the couple of years needed to really do full process characterization. So it's a truncated version. So then you don't really have, that full understanding of the design space of what you can and can't do with your process. And so again, going back to, if you can leverage past experiences, at a CDMO or another, consultant that can help advise you as to, what are the critical things, what are things you will regret not looking at and not, spending a little bit of time doing, it is time well spent.
Well spent. All right, we're wrapping up. We're getting close to the end. So we're going to rapid fire. I'm going to give them each, give one take-home message that you want to leave with the audience going away out of all of this conversation that you think, a kernel nugget that they can carry with them. And I'll start, well, she's on the phone. So I'll start with you, Philip.
I have my notes on the phone.
Oh, okay. Go ahead, because you already were, you're kind of there.
The one thing that I will say is that oftentimes when you go to a different manufacturer midstream in clinical development, you're doing that because you feel like the newer CDMO, the manufacturer, is providing something that your original one can't. And what we have seen is that too often in terms of comparability, they spend the time to do the comparability from one manufacturing site to the new manufacturing site, which you obviously have to do, but then it's go full next speed in terms of manufacturing, rather knowing that you're picking someone who has the ability to do better. So take the time once you've done the comparability from one site to the other to then start thinking about how you can do some process improvements, what they can suggest. And that does mean you have to do more comparability analysis, most likely just tier 1 testing and things like that. But don't just because you were able to transfer it from one site to another, just now go full steam ahead. Take the advantage of the fact that you're working with someone who can probably provide you with some improvements.
All right, Anna.
I think my nugget is communication. I think I said before, comparability is not just CMC. There's a cross-functional impact. I think you need to communicate cross-functionally and need continuous communication with the FDA as well in order to be able that you also consider the benefit risk of when to do the changes, if you should do the changes, and if not, what is the impact and the life cycle. So I think communication, I think, is where I would leave this conversation.
Process understanding, product characterization, and early engagement with FDA.
So 3 things very quick. So first, choose your manufacturing platform wisely. whether you use G-Rex or something else, but make sure that you have the end goal in mind. Second thing is, from an operational standpoint, set up your standards, and the standards is the law. And then the last thing is, Lean Six Sigma, continuous improvement, provide, the statistical rigors and a structure that you need to do comparability, or just to provide more stable operations. And the agency likes very much organizations that live in Lean and Six Sigma and continuous improvement.
That's great. And I'm going to do it before I get around to applause. So in the notes to this session, there was mention of two sides of the same coin. Here's my coin. It's comparability. All right, so I've got pre and I've got post. And if you're in development and you're doing this and you're going, what should I do, pre or post approval, you're too late. So you want to know ahead. I think they've given you all of the examples of what you need to know about how to make that and execute that. I want to thank my panelists. You guys have been great. You really dealt with me nicely. Thank you very much. And thank you, audience, for your questions. Really appreciate it.